In this video, I'm going to discuss about the bacteria Staphylococcus. Here is the content. I'll discuss about the taxonomy, morphology, diseases, and all others. So let's start. First, I will discuss about the taxonomy. Its family belong to the Micrococci family, and genus is the Staphylococcus. Species is the Staphylococcus aureus, Staphylococcus epidermis, Staphylococcus saprophyticus. And morphology, they are spherical in shape, like you see in the pictures, and spore is absent, capsule is also absent, motility is absent because there is no flagella, cilia are present, that's why motility is absent. Arrangement is grape-like clusters, and method of staining is the gram staining. It is required in this process, and it is gram positive bacteria. Next, about the disease, it causes superficial infections like pustules, wounds. Pustules means the small bumps on the skin like this in the picture, and also cause deep infection, toxic food poisoning, and toxic shock syndrome. And skin exfoliations means the removing dead cells will also occur. Respirations are through aerobic and facultative anaerobic respiration types. Next is about the cultural properties. They grow well on the ordinary cultural media, and temperature is about 37 degrees Celsius, and it requires pH for its optimum requirement is 7.4 to 7.6. In salty media, they form circular, convex, and pigmented colonies, and liquid media give opacity and become viscous mass. And in blood agar form colonies with beta hemolysis. And in nutrient media, nutrient agar colony produce pigments, and EX agar form colonies triggers mid backbone. And next is about the biochemical activities. It shows catalyze positive because it converts hydrogen peroxide to the water and the oxygen, so it shows bubbles. And oxidase positives because it produces indophenol blue from the oxidation of the dimethyl paraphenol diamine, and show coagulase positive where it will change fibrin to the fibrinogen. Next is about the antigens. Antigens, they have peptidoglycans, protein A, and teichoic acid. And about the virulence factors, they have microcapsules, teichoic acids. Enzymes are they have catalase, pectinase, DNase, and the coagulase enzymes are also present. Cytotoxins like leucine, beta hemolysis are present, and toxins, for example, enterotoxin A and B, enterotoxin and F, enterotoxin B and C are present. And about the pathogenesis, it develops as a result of the virulence of the bacteria and decrease its pores defense, as we know what pathogenesis is. And source of infection is the infected person and carrier patients. Mechanism and ways of transmission is from person to person through contaminated hand, fecal mechanism, and wound infection and vaginal flora. Next is about the sensitive organs like skin, vagina, and nasal passage, oral cavities, gastrointestinal tract, and all tissues. Immunity is a post-infection cellular and humoral immunity which is unstable in nature. And laboratory diagnosis is through specimens. We will take the specimens: pus, sputum, blood, urine, CSF, feces, food, and vomiting mass we will take. And methods are the bacteriological method of investigation, microscopy, and cultural isolations like pure culture isolation we will use. And for prophylaxis means how we will prevent through staphylococcal vaccine and toxoid, isolation and treatment, sterilization of the instruments, and the detection of the carrier among hospital staffs. And about the treatment, it causes vancomycin for Staphylococcus aureus and cephalozine and other nefcillin or oxycillin we will also use for the treatment. That's all about the Staphylococcus.